Hello everyone and welcome to the new session of the lecture series. And today we'll be solving the NCERT based multiple choice questions from the chapter Photosynthesis in Higher Plants. So we've already discussed some of the multiple choice questions in the previous lecture and we'll be solving and discussing the rest of the MCQs in this lecture. Alright, so let's start with the first question, which is the correct sequence of Kelvin cycle. Okay, Kelvin cycle is also known as C3 cycle. As the first stable product form is a 3-carbon compound. Now, Kelvin cycle takes place in 3 phases. Okay, first is carboxylation. Alright, first is carboxylation. During carboxylation, what happens? The carbon dioxide combines with RuBP, ribulose 1,5-bisphosphate, in the presence of the enzyme Rubisco. Okay, and what is formed? 2 molecules of 3-phosphoglyceric acid are formed. Alright, carbon dioxide contains 1 carbon. RuBP contains 5 carbon, alright, and 3-PGA contains 3 carbon, alright. That's why 2 molecules are formed. Now coming to the next step, which is the reduction phase. Alright, during reduction, what happens? The 3-phosphoglyceric acid which is produced is first converted into 1,3-bisphosphoglyceric acid. Alright. And then what happens? Here ATP is used, which is the product of the light reaction. ATP is used and ATP is converted into what? ADP. Okay. Next, what happens? 1,3-bisphosphoglyceric acid which is produced is, okay, this phase is known as reduction. Reduction is going on here. The acetic group is converted finally into the aldehyde group. Alright, 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceraldehyde. Okay, this is phosphoglyceraldehyde. Aldehyde group is present now. Alright, phosphoglyceraldehyde. Here what happens? NADPH is used and it is converted into what? NADP. Alright, this is also a product of the light reaction. Now, the last phase is known as the regeneration phase. During regeneration, what happens? There is regeneration of RuBP. Alright, and this ensures the continuation of the Kelvin cycle. Okay, and again, ATP is used here for the regeneration of RuBP. Alright, so we have to determine the correct sequence of the Kelvin cycle. So first option is decarboxylation, oxidation, regeneration. Not correct. Second is decarboxylation, regeneration, oxidation. Again, not correct. Then we have carboxylation, oxidation and regeneration, which is not correct. And the fourth option is carboxylation, reduction and then regeneration. So what should be the answer? Option D is the correct answer. Alright, now moving on to the next question, which is which of the following ratio is correct for the production of 1 molecule of glucose through 6 rounds of Kelvin cycle? Alright, first, okay, let's see what is glucose. Glucose is C6H12O6. Alright, this is what? This is glucose. Means it contains 6 carbon. Alright, and 6 rounds of Kelvin cycle are required to produce 1 molecule of glucose, or you can say 6 carbon dioxide have to be fixed. Okay, or carbon dioxide had to be fixed 6 times. Alright, now let's see the equation. First is the carboxylation phase, during which what happens? Carboxylation we have already discussed in the previous question. Let's take 6 carbon dioxide, which will combine with 5 RuBp. And what will they result? They will result in 12 molecules of 3-phosphoglyceric acid. Okay, 12 molecules are formed here. Now, these 12 molecules of 3-phosphoglyceric acid, okay, first is the carboxylation phase, second is the reduction phase. So 12 molecules of 3-phosphoglyceric acid is first converted into 1,3-bis-phosphoglyceric acid. Alright, and again here what will be produced? NADP, here ATP is used. Alright, how many ATP? 12 ATP will be used because 12 molecules are present here. Okay, 12 ATP are used. Okay, and it will be converted into 12 ADP. Alright, so this you have to note here in this step. 12 ATP are utilized. Alright, now coming to the next step, which is the conversion of 1,3-bis-phosphoglyceric acid to 3-phosphoglyceraldehyde. Alright, this is the reduction step. Again, 12 molecules are present. Alright, 12 molecules were produced here. So 12, okay. So how many NADPH will be used here? 12. 12 NADPH are used here. Okay. And NADPH will be converted into what? 12 NADP. Okay. And 12 molecules of 3-phosphoglyceraldehyde are produced. Okay. Now let's talk about the third phase, which is the regeneration phase. So what happens here? Now out of the 12, okay, 2 molecules of phosphoglyceride are used for the production of the glucose. Okay, out of this 12, 2 are utilized for what? For the production of glucose or carbohydrates, and 10 are used for what? 10 are used for the regeneration of RuBp. Okay, and 6 ATP are utilized in the regeneration of 6 molecules of RuBp. Alright, this also you have to note. Here 6 ATP are used. Okay, in this step 12 NADPH were used. Okay, so now let's look at the options. Okay, so we have to select the correct ratio here. So for 1 carbon dioxide they are saying 2 ATP and 2 NADPH. So this is not true because 3 ATP are used. Okay, if we talk about 1 carbon, alright, this was for 6 carbon. Okay, now we can write that for 6 carbon dioxide, for fixing 6 carbon dioxide, 18 ATP are used. 12 plus 6 is 18 ATP and 12 NADH. NADPH are used, okay, for fixing 6 molecules of carbon dioxide. So for 1 carbon dioxide, how much ATP will be utilized? 3 ATP and 2 NADPH. Okay, so this option is not correct. Second, 6 carbon dioxide, 18 and 12 NADPH. So this is correct. I've shown you how this is correct. 6, 12 and 18. This is also not correct, and then this is also not correct. So what should be the answer? Option B is the correct answer. Now coming to the next question, which is which of the following statement best support the fact that photorespiration commonly occurs in C3 plants? Okay. So what is photorespiration? Photorespiration takes place it is due to the oxygenase activity of the Rubisco enzyme, when Rubisco combines with oxygen instead of carbon dioxide. Alright. Then what it forms? 1 molecule of 3-phosphoglyceric acid and 1 molecule of phosphoglycolate. Alright. During the high oxygen conditions, okay, or high oxygen to carbon dioxide ratio, or during very high light intensity, alright, or during very high temperature, Rubisco combined with oxygen in C3 plants. Alright. So now let's read the statement, which is C3 plants don't possess trans anatomy. Alright. Now option B is C3 plants usually have high carbon dioxide compensation species. Alright. Third option is C3 plants are less efficient in photosynthesis, and fourth option is C3 plants are characterized by RUBP oxygenase activity under high oxygen supply. Okay. So which statement best supports the fact that photorespiration commonly occurs in C3 plants? What should be the answer? Option D. C3 plants are characterized by RUBP oxygenase activity under high supply of oxygen. Okay. So what happens? For photorespiration to take place, alright, I've written it in short. Photorespiration, first, high oxygen is required, high oxygen levels, or you can say high oxygen to carbon dioxide ratio is required. Then what? Then we need, during the high light intensity also, photorespiration is favored. And also during high temperatures also, photorespiration is favored. Alright. So now coming to the next question, which is during photosynthesis, when phosphoglyceric acid is changed into phosphoglyceraldehyde, which of the following reaction occurs? Okay, options are oxidation, reduction, electrolysis and hydrolysis. So we have discussed Calvin cycle in detail. Okay, so it is the reduction process, the reduction of the acidic group, okay, into the aldehyde group. So this is reduction. Now let's talk about the next question, which is number of carbons in the primary carbon dioxide fixation product of C4 plants. Okay, they are talking about the C4 plants here. Okay, and the options are 2 number of carbons, 3 carbons, 4 carbon and 5 carbon. So what happens in C4 plant? The acceptor of carbon dioxide is what? A 3 carbon compound known as phosphoenol pyruvate. Okay, which is a 3 carbon compound. Carbon dioxide contains 1 carbon. So what happens in the presence of the enzyme? Pep carboxylase. Okay. In the presence of the enzyme PEP carboxylase, oxaloacetic acid is produced, which is a 4-carbon compound. Alright. OA is oxaloacetate or oxaloacetic acid. Okay. So what should be the answer here? Primary carbon dioxide fixation product they are asking. Okay. So option C is the correct answer. Now moving on to the next question, which is the primary carbon dioxide fixation product in C4 plant? So here they are asking about the product, not the number of carbon. What is the primary product? Okay, so options are 3-phosphoglyceric acid, oxaloacetic acid, phosphoenol pyruvate, and ribulose 1,5-bisphosphate, RUBP. So we've already seen the primary product is oxaloacetic acid, which is a 4-carbon compound, which is the first, you can say, primary product or the first stable compound or the primary product or the product which is formed first. Okay, so that's why C4 plants has been given the name C4, because the primary product is a 4-carbon compound known as oxaloacetic acid. Okay, so option B is the correct answer. The next question is the site of photosynthesis in C4 plant is, first is mesophyll cell, bundle sheath cell, second option, both A and B, and fourth is cytosol. Okay, so again let's talk about the C4 plants. Okay, example can be maize, sorghum. Okay, so what happens in C4 plant? Carbon dioxide fixation occurs 2 times. Okay, first in where? In the mesophyll cell. In the mesophyll cells, where which enzyme is present? PEP carboxylase enzyme is present here. And the second carbon dioxide fixation takes place in the bundle sheath cells. Okay, where which enzyme is present? Rubisco enzyme is present. Alright. So, what should be the site of photosynthesis? Both A and B, both mesophyll cell and the bundle sheath cells. Okay, so option C is the correct answer. The next question is the primary carbon dioxide acceptor in C4 plants is, first is Rubisco, RUBP, ribulose 1,5-bisphosphate. Option B is phosphoenol pyruvate. C is PEP carboxylase. So PEP carboxylase is a enzyme. Okay, so it cannot be the acceptor. And fourth option is phosphoglyceric acid. Okay, so C4 plants, we have already discussed that carbon dioxide is accepted by phosphoenol pyruvate, which is a 3-carbon compound. Okay, and this leads to the formation of oxaloacetic acid, which is a 4-carbon compound in the presence of the enzyme PEP carboxylase. Okay, then what should be the answer? Option B is the correct answer. Now coming to the next question, which is fixation of carbon dioxide molecule through Hatch and Slack pathway requires an enzyme called. So what is Hatch and Slack pathway? It is basically the C4 pathway. Okay, C4 pathway is also known as the Hatch and Slack pathway. Okay, now which enzyme is involved here? They are asking. First is PEP carboxylase, second is Rubisco, then we have RUBP carboxylase, and the fourth is oxygenase. Okay, so what should be the answer? We already know that the Okay, let's talk about C4 pathway again. So initial or the primary carbon dioxide fixation. Initial or the primary carbon dioxide fixation, okay, requires where, first of all, it takes place where? It takes place in the mesophyll cell. Okay, and which enzyme is involved? PEP carboxylase. Okay, now secondary, we know that the secondary or the second carbon dioxide fixation takes place in the bundle sheath cell. Okay, where Rubisco enzyme is present. Okay, so what should be the answer here? Answer is PEP carboxylase. Okay, so here don't get confused between this option B and option A. Okay, they are talking about the primary carbon dioxide fixation here. Okay, if they have mentioned in the question that both primary and secondary carboxylation, if they are asking about the primary and secondary carboxylation, then you have to mention the both of the enzyme. Okay, now moving on to the next question, which is bundle sheath cells, okay, are rich in which enzyme they are asking. So are rich in Rubisco, are rich in PEP carboxylase, lack Rubisco, or lack both Rubisco and PEP carboxylase. So now we already know that bundle sheath cells contains which enzyme? Rubisco. Okay, which is involved in the C3 cycle. Alright, so this was very straightforward question. Now moving on to the next question, which is in the leaves of the C4 plants, malic acid formation during carbon dioxide fixation occurs in the cells of, okay, again let's talk about the C4 pathway or the Hatch and Slack pathway. So what happens initially? Carbon dioxide is accepted by phosphopenol pyruvate, which leads to the formation of oxaloacetic acid. Now this oxaloacetic acid cannot enter the bundle sheath cells. So these are bundle sheath cells here. Okay, here we have the mesophyll cells. Alright, so this is happening in the mesophyll cell, this reaction. Okay, now oxaloacetic acid cannot enter the bundle sheath cell directly. So first it is converted into what? Malic acid. Okay, first it is converted into malic acid. I'm writing it in short form, malic acid. Now this malic acid can enter the bundle sheath cells. Okay, so malic acid is produced where? It is produced in the mesophyll cells. Okay, then what happens? Malic acid is converted or decarboxylated. Okay, and carbon dioxide is released plus pyruvic acid. Pyruvate is formed. Now this carbon dioxide can be utilized in the production of the carbohydrates by the C3 cycle. Okay, here Rubisco enzyme is present in bundle sheath cells. Okay, so what should be the answer here? Options are bundle sheath, phloem, epidermis and mesophyll. What should be the answer? Option D is the correct answer. Malic acid is produced in the mesophyll cells. The next question is, by looking at which internal structure of the plant can you tell whether a plant is C3 or C4? Okay, so C4 plants. Let's talk about the C4 plants. So C4 plants, okay, they are adapted to dry tropical condition. Okay, or you can say arid region or semi-arid region. So what happens? They show a unique leaf anatomy. Okay, they show a unique leaf anatomy, which is known as what? Which is known as Cran's anatomy. Okay, so this is a adaptation of the C4 plant to high temperature. Okay, so in Cran's anatomy, what happens? The mesophyll cells, first of all, there are two types of cells. First is mesophyll cell and second one is the bundle sheath cells. Okay. Now, and there is a spatial separation of the carbon dioxide fixation. One takes place in the mesophyll, other takes place in the bundle sheath cells. So, in Cran's anatomy, what happens? The mesophyll cell are clustered around the bundle sheath cells. Okay. And what does it do? It forms a wreath. Okay, or you can say a ring. Okay. What does crans means? Crans means a wreath. Okay. So, on the inside we have the vascular bundles. Okay, so these are surrounded by the bundle sheath cells. Alright, which contains large chloroplasts. Okay, and which are agranal. Now, these bundle sheath cells are surrounded by the mesophyll cells. Okay, or you can say mesophyll cells are clustered around the bundle sheath cells. So, these ones are mesophyll cell. Okay, these ones are bundle sheath cells. Alright, so this is known as the Cran's anatomy. So this is a survival strategy of the or adaptation of the C4 plants to high temperature. Okay, and because of this, there is a separation of the carbon dioxide fixation. Alright, so now let's look at the option. Cran's anatomy is the first option. Distribution of mesophyll cells. This is also true. Mesophyll cells are arranged or they are clustered around the bundle sheath cells. Okay, so this is also a unique feature of leaf anatomy of the C4 plants. Okay, third option is bundle sheath cells only. Not correct, and both A and B. So option D is the correct answer here. Both A and B. Alright. The next question is, in C4 plant, the process by which C4 acid is converted into C3 acid in the bundle sheath cells is known as, okay, first option is carboxylation, second is degeneration, C is reduction, and then fourth option is decarboxylation. Alright, so C4 acid. So which C4 acid is formed? Okay, again, let's talk about the C4 cycle. Carbon dioxide combines with phosphoenol pyruvate. Okay, leads to the in the presence of enzyme PEP carboxylase, leads to the formation of oxaloacetic acid, which is the 4-carbon compound. Oxaloacetic acid converted into malic acid. Okay, this malic acid then enters the what? Bundle sheath cells. Okay, here bundle sheath cells, here we have the mesophyll cells. Malic acid enters here. This malic acid is then decarboxylated. Means there is a removal of carbon in the form of carbon dioxide. Here carbon dioxide is released, which is used where? In the C3 cycle. And what is formed? Pyruvate is formed here. This pyruvate goes back to the mesophyll cells and it gets converted into phosphoenol pyruvate. Okay. So, what should be the answer here? Decarboxylation. Okay, there is a decarboxylation of the C4 acid releasing carbon dioxide, which is used in the C3 cycle for the generation of C3 acid, okay, which is 3-phosphoglyceric acid, okay, which is a 3-carbon compound. The next question is, which of the following pair is not correctly matched? Alright, C3 plant, maize, C4 plant, Cran's anatomy, so this is correct. C option, Kelvin cycle and phosphoglyceric acid, this is also correct. Hatch and Slack pathway, oxaloacetic acid, this is also correct. Okay, Hatch and Slack pathway is the C4 pathway. Now, what should be the answer here then? Option A, this is a mismatch. Maize is what? Maize is a C4 plant and not C3 plant. Okay. The next question is, in C4 plants like sugarcane plant, 14 carbon dioxide is fixed into malic acid. Which of the following enzyme is used to fix carbon dioxide? Okay, so here 14 carbon dioxide, they are just, okay, confusing you. We have to just answer which of the enzymes from the below is used to fix carbon dioxide in C4 plants. First is fructose phosphate, not correct. Ribulose biphosphate carboxylase. Okay, then we have phosphoenol pyruvic acid carboxylase. This is correct. D option, ribulose phosphate kinase, not correct. So what should be the answer? Phosphoenol pyruvic acid carboxylase, also known as PEP carboxylase. Alright, which is involved in the primary carboxylation. Alright, so this was the end of this lecture. Thank you very much for listening.